is the Chief Precision Medicine Officer at BPGbio. Dr. Kiebish oversees omics, biomarker, assay development, and informatics teams at BPGbio. Michael and his team have pioneered novel systems, medicine strategies for biomarker, pharmaceutical, and theranostic development to ensure that patient biology is at the forefront of drug discovery. Through the implementation of artificial intelligence platforms, big data integration, population health strategies, and omics technologies, the platform and translational science group overseen by Dr. Kiebish synergizes BPGbio's efforts in improving patient care and guiding drug discovery, as well as clinical trials of informed molecular insights to improve translational outcome. A very warm welcome to you, Michael. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And now, without further ado, I would like to hand over to Michael to begin his presentation.
Thank you, George, for the warm introduction. Good afternoon and good morning, everyone. Really an honor to be here and looking forward to really highlighting BPGbio's AI and biology-driven approach. Covering the landscape of BPGbio's approach, we really have pioneered a biology-first AI-driven drug discovery platform. Now, this approach really integrates longitudinal, clinical, molecular, and patient data, and it embodies multi-omic technologies. And again, I'll be diving deeper into the type of multi-omic biological technologies we'll be actually utilizing and harnessing into domain-specific AI, particularly Bayesian causal inference approaches, along with computational chemistry approaches, doing fragment-based screening, high-throughput computational methodologies. Now, this is really embarked on creating a wholly owned clinical stage pipeline, really encumbered on BPM 31510, which is really a way to modulate metabolic activity on cancer and dysfunctional mitochondrial disorders, which we've moved into phase 2 clinical trials for GBM. Also, we've really developed a novel program for protein degradation and protein homeostasis using molecular glues and targeted scaffolds for E2-based protein degradation. The current landscape of protein degradation of molecular glues is heavily focused on VHL and Celebron. However, we have determined, and I'll go into more detail, an E2 mechanism to actually engage and actually overcome some of the challenges with E3s. And as you see here, this is our new facility, actually highlighting cutting-edge technology with this integrated omic approach. Now, moving to really the four pillars of the NIE interrogative AI approach, integrating biology through discovery to validation. I think it can't be emphasized enough that the sample generation and the sample of origin where the biological information comes from is at the foremost criticality to actually translating into actual therapeutic products or diagnostics. We've embarked in the past 14 to 15 years in collecting longitudinal samples from patients, whether they be cells, PBMCs, buffy coat, serum, plasma, urine, saliva, a range of different biofluids, which allows us to really stitch together actionable biology. We've also used those cells to interrogate in a culture room to actually identify targets and mechanisms as well. So this has been critical, working with longitudinal samples and also working across industries, working across academic centers and geographies in different diverse ethnicities to actually understand human biology at its core. But this is a starting point of discovery. Further, with multi-omics means a lot of different things to different people. For some, it means genetics and transcriptomics. For us, it's really the phenomics and expososomics, what human biology is exposed to and how we can actually capture the changes that we see during disease phenotype. Now, this can occur at the proteome, it can occur at the post-translational nature of proteins. And when you think about the proteome, it can range from 8 to 12,000 proteins. From PTMs, they can be phosphorylation state, they can be ubiquitination, acetylation, methylation. There's 200 PTMs that exist, and this can basically sum up to tens, to hundreds of thousands, to even millions of modifications on proteins. This can be the lipidome and metabolome, beyond just the transcriptomics and genomics. Further, spatial omics technologies, which we have been engaging, is really critical as well. Before, years ago, 5-10 years ago, we'd grind up a tissue and understand omic technologies. Now we're getting down to the single cell level, how cells are communicating, because biology really is a society of cells, society of tissues, and how that is exposed to the environment to initiate disease. Further, the third pillar is really having wet lab validation technology in-house, actually interrogating the biology in-house, doing target validation, target MOA, screening activities using novel enzyme activity, whatever the actual assay might be. But further, the fourth is really stitching this all together with a Bayesian approach, an unbiased approach, which actually generates novel insight. It's going beyond what really is a big push in generative AI and machine learning neural networks. This is taking data with no a priori knowledge and actually causally integrating it. And it can be very diverse data sets, molecular data, real world data, and beyond. So this is really critical to translate this into target discovery, mechanism of action, biomarker development, asset screening, drug repurposing, and broad epidemiological studies, which we've embarked on and published quite a few. So this integration is really key, these four pillars for the platform. Now, how does this Bayesian approach really look? Unfortunately, due to technical difficulties, I'm not able to show the video, but I think this is just a snapshot insight into how Bayesian novel insight generation actually works. When you take tens of thousands or hundreds of thousands of variables off of thousands of samples, you want to understand how each is connected to two or three different entities. Now, this occurs in a vector algebra manner. Two or three things come together at a time. They have a statistical correlation, and then they are essentially combined into smaller networks. Bit by bit, these smaller networks are actually integrated into massive networks. And these in informatics are called hairballs. And this is done again and again, hundreds to thousands of times, until we see continuity and homogeneity in the actual network directionality and what is a high confidence network, what are high confidence connections, all based on pure biological knowledge. And I know in biology, we're trained to always follow the things that are the greatest changes. However, in Bayesian, we are allowed to see subtle changes actually have a very strong signal away from the noise. Thus, measuring biology very quantitative can actually indicate things that might have a 10 or 20% change, but that impact is really the deciding factor in biology, whether a disease will occur or not, and also what should be targeted. Now, another key aspects of a Bayesian approach from an AI, how we actually go from biology to an AI translational insight, is really defining what is connected, what is the interactome of a normal or non-treated and actually treated or disease situation or a disease impetus. How are things connected differently? How are the highways actually connected in biology? And we can actually run delta networks to understand the differential between disease and normal. We can understand, really, as biologists, as humans, we like to not understand a million things. We like to focus it and dwindle it down to about 10 things that can really be interpretable. And a lot of times I call this trickle-down biology. And why I call it trickle-down biology is because you're trying to find the one or two or 10 nodes that are affecting not just one or two things, hundreds of things. And in those nodes are affecting thousands of things. So this kind of network topology analysis and vector algebra really allows biologists, translational scientists to focus on really the important driver. What I will say is this kind of novel discovery does come up with a lot of known biology. And it's really exciting to find, whether in neurological disorders, finding ApoE, the known genetic association with Alzheimer's, come up as a top node. Other neurological disease and cancer, major drivers of cell cycle, coming up independently. Seeing proteins that are connected in networks come up independently is really critical. And what we filter this down is to, again, the top 10 list, which allows us to understand WIPO, is there IP filed on it? There are a lot of publications, what is known and what is unknown, and what are real key drivers of disease, whether a cancer cell is dying or proliferating, whether a neurological cell is dying or being dysfunctional, whether a patient has a clinical outcome or adverse event, what is driving that? So really a lot of different biological and clinical translational questions can be addressed by this approach. Now, another fundamental thing in the field is really to understand that biology is diverse, and we need to really go beyond our DNA. Even though DNA, whether it be genomics, SNPs, transcriptomics, is really critical, it is part of our evolutionary nature. It defines our ethnicity and our genetic heritage and risk for disease. However, the vast majority of diseases are age-related. They are connected to the expososome and the phenome, what phenotypically is driving our molecular phenotype. And that is where we need to focus. Is it a phosphorylation modification? Is it a acetylation or ubiquitination or methylation state in proteins that are circulating in tissues that are changing us? Because our evolutionary history is important, but our actionable biology connected to the phenome and expososome is what's driving disease in age, in all these metabolic, inflammatory, neurological, and oncological diseases. That's where our biology begins, and that's why we focus heavily on more adaptive omic technologies beyond genomics. Even though genomics is important, we can't be myopic in thinking that's the only thing driving the nature. And further, biology isn't in complexes. Proteins exist in complexes. And transcriptomics has been shown to not necessarily always change with complex formation, but proteomics has. And all these other omics that also coalesce with these protein changes and complex changes also been shown to connect it. So we are actually seeing dynamic biology where we measure these other omics beyond genomics. Further, we've actually extended a lot of partnerships, one being with the U.S. Department of Defense, U.S. Department of Energy. Solving major pharmaceutical and translational development efforts cannot exist alone. We have to partner with key individuals who are like-minded and have resources to really move the needle forward. With the U.S. Department of Defense, they were very visionary 25, 30 years ago in creating biorepositories, biobanks, creating tens of thousands of samples, whether they be tissue, plasma, serum, other types of biofluids, to actually have longitudinal history, have good pathological characterization samples. Now, this is needed to actually start the discovery, as I mentioned before, on a biorepository. Other international efforts like Genomics England and All of Us program in the US have also started that effort. But obviously those programs are now decade plus into the working. So it's really critical to have this insight, and the US Department of Defense has been that way, and we've partnered them to actually develop breast cancer and prostate cancer diagnostics using our biology first AI approach. Further, BPGbio has recognized that computational power is critical, and we work with the U.S. Department of Energy at Oak Ridge National Laboratory using their Summit and Frontier computer to actually engage our algorithm at a quantum computing scale to allow Bayesian insight to gain a really complex data sets. We've used this with COVID, real-world data, as well as some of our target discovery. Further, they have a really advanced molecular biophysics program, which allows us to screen millions of compounds in extremely short time frames to actually translate that into hits to leads and assets in clinical development. Those partnerships are absolutely essential. Now, focusing how we are using our biology first AI to focus on key indications, especially affecting age-related disorders. One key area is mitochondrial medicine, and the other is protein homeostasis. For mitochondrial medicine, we have a clinical asset, BPM-31510, which is in clinical stages of development for glioblastoma and pancreatic cancer, really highly metabolic, aggressive cancers. Additionally, we're developing BPM-31510, which is a novel formulation of oxidized CoQ10 for CoQ10 deficiency, electron transport chain disorders, potentially Friedreich's ataxia, and other key indications that are basically known to have mitochondrial deficiencies, ROS associations, and energetic failures. Now, shifting gears to protein homeostasis, our AI approach has identified a novel E2 mechanism. So in protein degradation and proteasomal degradation, you have E1, E2, and E3, and other components come together to form the proteasomal components for degradation. Most drugs are being developed currently using E3 mechanisms such as VHL and Celeberon to actually target protein degradation. We found that E2 is a novel scaffold in a modified E2 could actually be engaged to utilize, and this E2 actually has a UBA domain, which I'll talk about more. They can actually do the processivity function, and it can be engaged as a molecular glue for processivity on, or another component actually processivity off. So we are really capitalizing on what the AI has driven into this biological insight, which is really harnessing both these platforms to tackle age-related diseases and other diseases that have a huge impact on society. As I mentioned, our pipeline currently is encumbered by glioblastoma. We're currently in a phase 2 enrolling trials in multiple different sites for pancreatic cancer, for primary CoQ10 deficiency, for direct replacement of CoQ10, or topical formulation actually engaging wound healing, which I'll talk about more in epidermolysis bullosa, or sarcopenia, which has a clear deficiency in CoQ10, which is leading to energetic failure in muscle and muscle weakness. For actually a very interesting mechanism, which is wholly owned, BRG-399, which targets tubulin polymerization in oncology, but also in impacting neutrophil chemotaxis and degradation. And so that is really critical for either gout or cardiovascular health. Additionally, this E2 mechanism has identified Huntington's disease as a major target, where we're targeting kind of eliminating Huntington aggregation. And further, this protein degradation platform, E2 base, is using multiple targets in oncology and other diseases, whether inflammatory or also associated. So really a robust, internally developed pipeline, all driven by AI. Now shifting gears to AI and drug discovery, we all know the landscape is quite crowded. We've been doing this for 15 years, really focusing on a pipeline and indications. And AI really is a tool that has to be utilized. We have multiple phase 2s in clinical trial. And you can see the landscape. Some companies are just getting started. Some companies are really in the preclinical phase. Some companies have dozens of trials ongoing, really trying to show their AI could move the needle. BPGbio is really focused on really key understanding where our indications fit, where they can be expanded, rather than having too many programs, because really AI is a tool and it has to be better and it has to be focused, it has to be proven. If the drug works and the mechanism works, more indications will come rather than having too many programs to really take this field off. But there is a crowded field, but it really is what kind of AI has proven to have translational utility for patients. Now, again, highlighting AI first and biology first. I know a lot of companies, AI companies, really just use publicly available data sets and make the assumptions around that. They don't have wet lab technologies. They don't have the validation. They don't do their own omics in-house. They don't have assessment of the quality of the omic data or publicly available data. And they're not asking the right biological question up front and then getting the data or generating the data. We feel that the question needs to be up front. The biology needs to be up front and discovered and decided upon. You have to do your own omics analysis. You have to understand the quality and the variation of data. You have to capture the diversity of data in the same exact sample. You can't be running one type of omics on another set, another type on another, and trying to merge together. So the data from the wet lab is essential. Having discovery done on high quality samples with key questions that are powered is essential. Doing validation on publicly available data sets is okay, but discovery has to be on high quality data. And without knowing the pathology, without knowing the insight of the sample in the public, not knowing the quality of the platforms, how things are being integrated, it is a major challenge deep down. And is there enough sufficient publicly available data that can be stitched together with generative AI? If generative AI doesn't know the quality, doesn't know the question that was originally proposed. So this is really essential to the change in the field as a whole and what is being embarked upon and what's being promised within the different AI technologies in the field. Now, good AI starts with good data. I can't emphasize it enough. I mentioned longitudinal data, essential tissues, cells, biofluids, longitudinal clinical data, data dictionaries. How are patients actually being defined? And how clinical measurements are actually being defined is really so important. Further, the idea that patients, that one sample is a snapshot in time, it's not really reflective of the biology. We all know our biology changes, our clinical phenotype, our health changes daily, hourly. And so the more samples we have of the same patient, the more diversity of samples, because everyone has comorbidities, concomitant medications, different diseases, different risk factors, that allows for understanding the directionality of our health, of our biology. And that couldn't be emphasized even more. And further, I know I mentioned it again and again, what is multi-omics and what people believe is important. The range of multi-omic technologies, even glycomics, is becoming so important to understand the society of cells, the integration between the immune system and our own health, the microbiome. Everything has to be captured to understand what good data can be layered into actionable insight. We cannot interpret billions and trillions of data points as humans. So we're using this AI as a tool to simplify this and integrate it in a very palatable, communicable way to prioritize in those 10 or 20 things that are actually actionable. Now, this again comes from using the biorepository data, generating different models, and I can't emphasize it enough. Partnerships are key. Academic, industry, government. We cannot do this alone. We cannot we can't access all the samples we need. We need to work with key institutions that have been foresighted enough to have 10, 20 years of longitudinal data, uniform clinical record systems, government systems that have been able to do this, understanding, characterizing the diversity of different diseases. How are they uniformly characterizing their neurological state? This can't be emphasized enough because that's how AI can actually have the actual insight. Further, we need that ethnic diversity. We need to do geographies where countries have different Hispanic, Asian, African American origins, different geographies to integrate the diversity and exposomics that actually occurs that allows us for disease to penetrate and actually have its impact. And just highlighting again, causality versus correlation. Two of my favorite examples of correlation based on data alone is the example when the Greek economy started to fail and Facebook, and at the same time, Facebook users picked up. If you look at this graph, you would see a perfect correlation that Facebook caused the collapse of the Greek economy. Now if we look at biological data, that would be completely false. Because if data was just having this correlation, we would make the assumption and actually target Facebook, causing a collapse of the economic system. Further, one of the best examples of visual imagery of causal AI is this cat right here causing that roof to actually collapse. Because if you just look at a snapshot in time and data alone, you would make that assumption. However, we would not know that a car hit this roof or some other kind of structural impact hit this roof. That snapshot in time, we're correlating, we say the cat is causing it. When a car crashes, we don't know, if you assume that the car is the reason for the car crash, we don't know if people were texting, we don't know if there was an oil slick, we don't know what happened. And if we went after the car industry, we wouldn't have cars. But understanding causation versus correlation is absolutely critical to making decisions, especially for biology. And causal AI models have to have directionality. They have to take very diverse factors, clinical, phenotypic, psychological, any information capable of being imputed, dynamic or binary, has to be imputed in a way to understand what are the primary drivers, what are things going outwards that are actually effects of what could be biomarkers. So causal AI in therapeutic development is not possible without patient biology, and all the data comes around population health, and that is really essential. And further, AI has been promised to be the end-all solution. It is not. It is an enabler. It's not a replacement. We need biologists. We need pathologists. We need data scientists. We need clinicians, all to ask the question, all to work together on what kind of data needs to be inputted and what do we want to get out of it, and let AI compute those billions and trillions of data points into that palatable 10-component factor. There's a reason why phone numbers are 9 or 10 digits, because human mindset can understand 10 things at a time and remember them. So we need to distill it down to the 10 most important things, 20 most important things to actually integrate and translate into it. So we're not trying to replace biologists or clinicians or scientists. We need to work together, and it is essentially a tool. It can't be promised beyond that. And for us, we've used AI to develop, as I mentioned in the pipeline before, for treatment for EB, discovering that skin is the largest organ, which we know, and that metabolic and anti-inflammatory activities, especially from BPM-315 on a topical, could have benefits in EB. And we've seen an 81% response rate and 24% complete histological closure in a disease which has chronic wound healing. Skin falls off. They have wounds for life. And actually seeing the wound closed has a huge impact on patients, their families, and beyond. Second, knowing that glioblastoma and pancreatic cancer is a massive metabolic disease, that BPM-315 can re-educate the tumor and actually make normal cells healthier is critical. And that's why we're really pushing a phase 2 clinical trial in GBM. We actually presented data 2 to 3 days ago in Barcelona at ESMO, highlighting the mechanism, highlighting the progress in the clinical trial. And further, sarcopenia. Sarcopenia is known, and AI biointelligence has guided us to knowing that CoQ10 is actually deficient. It's a driving force why muscles are weak. And actually delivering BPM-315 oxidized CoQ10 back can actually restore potentially restore mitochondrial function and the key hallmarks of the disease. All of this has really been critical to actually moving this needle forward. And finally, actually discovering an E2 platform, an E2 target, de novo from biology, which after diving into it further, actually has an UBA domain, can actually function as an E3. And then we realize more that E2s can work together beyond E3s to actually work through this mechanism. And at first pass, you would think, well, E2s don't have a lot of great pockets for druggability. However, when they're ubiquitous, when they're charged, they actually open up novel pockets. So this biology approach has been really highlighting through biointelligence and AI-driven and biology linking. This is a key target in cell death, proliferation, and other mechanism controlling cell activities. And by looking at it in a very diverse way, PTM modified, we can actually increase efficiency and potentially overcome major other factors of resistance that E3s potentially have, because there's such a dependence on Celebron and VHL that the other targets, other mechanisms, other scaffolds need to take place to actually engage. So this is really critical to this development and this strategy for drug development and using biological intelligence. So as I mentioned, putting biology first and putting AI first can actually speed up discovery, can actually highlight novel biological pathways, which are critical, can actually de-risk patient selection optimization for therapeutic indications. As I mentioned, GBM in pancreatic cancer, highly metabolic tumors that would be targeted. They can assist in clinical design, aligning with adverse events association or response signatures, capturing the diverse biology that's connected to the target using these omic technologies. And they can actually improve the predictability of clinical trial outcome because it's data-driven, it's biology-driven. And we forget that a cancer patient is not just a patient with cancer. They have components of the liver, other diseases, and we have to understand patient biology to improve and optimize the selectivity in clinical trial development based on that patient biology. So it can make a drug discovery faster in development, but it is only a tool. It is not a black box which is going to give the golden goose. It's going to be used to make more informed decisions. It will make it faster and hopefully better. And so hopefully this kind of covered the broad scheme of BPGbio's approach and how Bayesian technology and also biology can really streamline us and integrate it for full discovery. And I definitely appreciate everyone's attention on this afternoon and morning for them. And we'll be happy to take any questions in chat.
Thank you so much, Michael, for that incredibly fascinating presentation. We really do appreciate you sharing your expertise with us today. As with all the other talks, we're likely going to have lots of questions come in from the audience. And I can see that we have received some questions already, so I'll move straight onto the Q&A now. So Michael, your first question is, you mentioned that AI has been overhyped in its capabilities for drug discovery, but can you elaborate further on the specific challenges and misconceptions that the industry currently faces?
I think AI has been basically put out as a solution for everything. I think different AI technologies are going to be applicable to different aspects in drug discovery. I think they can be tailored based on the data, whether AI is really good to actually discover a novel kinase inhibitor or other tool. But I think we have to be very open, anyone working in AI, what it can do and what it can't do. And that is critical. And what kind of data is important and how they're actually utilizing it. It cannot be a black box. It has to be a white box where we can see into it and we can ask the questions. So I think that's been the challenge as an industry. There's been a massive hype and not an appreciation that it's a tool. It's not an end-all to be-all solution, and that we need all the components of drug development, part of it, asking the right questions to allow for it to actually output something actually useful.
That's wonderful. Thank you so much, Michael. We do have plenty of questions to go through and plenty of time left for this Q&A session. So moving on to your next question now. You mentioned that obviously this is an incredibly huge area and there are many key players involved, but can you expand on how your approach at BPGbio differs from other AI-driven drug discovery models, and why is it, I suppose, more effective?
I think one challenge in that is people don't openly talk about what their AI is. Is it just advanced statistics? Is it machine learning? Is it neural networks? What type of algorithms they're using, because they're always proprietary? What generative AI they're actually using. And so that is a challenge to what part of the drug discovery process they're actually using it for. I mean, there's 10 or 12 major pillars of drug discovery, whether it be target ID, patient selection, clinical development, actual computational chemistry. And there's 200 different components of actual drug development process. So by people not being able to openly say where they're actually affecting it and using it, or they're not using it, is kind of a challenge. I think our approach has been the Bayesian approach for target discovery and patient selection based on causality. And we clearly use a different approach for computational chemistry and other kind of machine learning approaches. So I think it really comes down to how many AI companies have moved things into phase 1 and phase 2. Have they been able to explain the utility of their AI and where they're using it? And have market differentiation occurred based on that and how broad their AI could be used or can be used in this specific indications, whether it be inflammation or certain kind of targets?
Another fantastic answer. Thank you so much, Michael. Now, how do you envision AI and biological understanding converging in practical terms? I know through your presentation you discussed a few case studies of where AI has been implemented, but are there any other current examples where this integration is proving to be highly successful?
I think a lot of algorithms in digital AI and actually understanding further how the data can be translated into more actual products and algorithms is really critical. I think diagnostics too. Diagnostics has been a real challenge because usually discovery occurs in a couple hundred patients or a couple other individuals, and then they try to approach it to a broader audience. Failure rate is really high. Whether those markers are discovered by correlation or causation can be helpful. I think in diagnostic development, you always want to use 2 or 3 approaches to actually refine whether you would move a set of diagnostic biomarkers forward or not, because one technology might not give you the right answer. So combining different analytic and AI solutions will. There's been a lot of success stories of actually defining a target and repurposing drugs from an AI perspective. That clearly is a kind of a loophole in the conventional drug development standpoint, but I think it kind of, again, highlights that drugs can be repositioned that already went through the pathway, and then defining a better indication from AI is also helpful.
Fantastic. Thank you, Michael. We still have time for several more questions before we close out this session. Now, we've had a comment from a member of our audience who has stated, obviously, there's many advances in precision medicine that are ongoing, especially with cancer. And there are obviously robotics which can be used to treat cancer and kill cancer cells. Do you think that this AI will be able to provide a better treatment option than using these robotics?
So I'll see if I can interpret the question: robotics from a discovery standpoint, from actual therapeutic, or actually more. So I'll approach the question from robotics to actually discovery. Yes, robotics can actually speed up the amount of screening, the amount of experiments that can be done. That can be merged with AI to get more data. Sometimes more data points are not as useful as broader data points. So I think that's really the convergence of the type of data being collected and how much screening and how much high throughput needs to be done. So I think converging actionable data versus volume of data is really the key thing when robotics come into mind. We need to collect data. We need to be uniform. Robotics is really good at precision, but it comes down to the data we each want to get, how much data is actually necessary, and what is actionable to really refine what is killing a cancer cell and protecting a normal cell, and what is that biology, because it has to go back into a human anyways. So actually analyzing in a dish doesn't always translate, but understanding what kind of data and how much volume of data is from the robotic experiment is kind of important to define. Hopefully that addressed the question.
That's fantastic. Thank you, Michael. I believe it definitely must have answered the question, and thank you for the expert answer. We have time for just a few more questions before we close out this session. So moving on to the next question now. Can you provide any sort of specific examples of how integrating patient biology with AI has led to successful outcomes in drug development at BPGbio?
I think for the examples we've had here, GBM actually seeing the kind of enrollment we're looking at glioblastoma, now that we're first-line therapy, knowing that glioblastoma is a highly metabolic tumor, how we're giving the drug, how we're dosing it, and how we're combining it has given us confidence that our Bayesian approach is really guiding us in the right direction. I think Right now, some of the mechanistic studies that we're doing preclinical in BRG-399 in terms of inflammation and neutrophil biology and kind of tubulin polymerization has guided us into a really strategy to move toward an IND, which is really critical. I think that the number one point I think everyone in the AI industry can agree upon, true validation of AI only occurs when a drug is approved and actually goes to market and has longitudinal effect that is done again and again. And I think that's the challenge with the industry, is that drugs de novo to approval is still a work in progress. So I think that is something we're really striving for and really appreciating, that we're using this tool to make that final leap to getting it all the way to approval based on all the data we're imputing into the AI.
Fantastic. Thank you, Michael. And we have time for one final question today before we close out this session. And what a better way to round out this session than what do you see as the future of AI in drug discovery and development? And I suppose, how will this field continue to develop?
I think the future, it will be a merger of AI technologies. It will be a combination. I think the complexity and volume of biology we're measuring now is opening up new doors. We appreciate, 10, 20 years ago, spatial omics, actually 5 years ago, spatial omics was not where it is now due to the instrumentation and the algorithms. Circulating tumor cells, fragmentome, everything about biology that we're measuring is going to require higher computational power and also require combinations of AI tools. So, as much as I would like to predict the future, I definitely see that the future really is a merger of AI approaches, not just one, not just generative AI or machine learning. It's a merger, and it has to handle the volume of data, petabyte, gigabyte, whatever volume of data it is, and how it can computationally streamline that analysis in a very uniform, simple way to actually, again, go down to 10 or 20 informed things to actually produce products that actually impact patients.
That's a wonderful answer and a great way to round out this session. Thank you so much, Michael, but unfortunately that is all that we have time for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. And as with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. So that just leaves me to say a massive thank you once again to Michael, and to everyone in the audience. We'll be back soon with the next talk.
Thank you.